Meiosis, a cornerstone of sexual reproduction that ensures genetic diversity by generating unique gametes with novel genetic combinations in multicellular organisms. It happens through the separation of homologous chromosome and genetic recombination during the specialized cell divisions of meiosis I and meiosis II. Are you ready for it? Meiosis is a process started by meiosis I, followed by meiosis II. In meiosis, there are certain basic units that you have to know to help you keep on track in this video. And these are what you're gonna know so that you are not getting confused in the following video. In SPM level, we have learned meiosis I is divided into prophase I, metaphase I, anaphase I, and telophase I, but in matriculation, prophase I includes 5 more stages, which are leptotene, zygotene, pachytene, diplotene, and diakinesis. Now let's go deeper and precisely towards the world of meiosis. In prophase I, chromatins become tightly coiled, thickened, condensed, and become visible. Synapsis of homologous chromosomes forms bivalence or tetrad. Crossing over occurs between the non-sister chromatids of the homologous chromosomes to exchange genetic materials. The nucleoli and nuclear envelope disintegrate and spindle fibers are formed. Now, let's discover the second process in meiosis I, which is metaphase I. In this phase, homologous chromosomes randomly aligned at the metaphase plate. The chromosomes of each pair of homologous chromosomes face different poles, and they are attached to the spindle fibers at their centromeres. Next, let's proceed to anaphase I, which is a splitting phase. Spindle fibers contract and shorten, which separates the homologous chromosomes. Each chromosome moves to opposite poles of the cell. Each chromosome still contains a pair of sister chromatids. In the last phase of meiosis I, telophase I, chromosomes arrived at opposite poles of the cell. Cytokinesis is followed up that acts as a chainsaw, which split the parental cell into 2 daughter cells with new genetic combinations. And remember, each daughter cell is now carrying half number of parental chromosomes. Till here, some people may be confused. What's the difference between spindle fiber and kinetochore microtubule? Sometimes we call the spindle fiber as kinetochore microtubule when it connects to the centromere of chromosomes, or we will call it a non-kinetochore microtubule when it overlaps the metaphase plate. Now let's head into the world of meiosis II. The process in meiosis II is similar to mitosis, but the chromosome number differs from mitosis. In prophase II, the chromatids shorten and thicken again, and new spindle fibers appear. Each chromosome aligns at the equator plate attached to the spindle fiber at centromere. Then, in anaphase II, spindle fibers shorten and contract. This causes the centromeres to split. Sister chromatids separate and move to opposite poles. Each sister chromatid now is called a chromosome. Lastly, we will see the changes in the number of chromosomes and DNA during meiosis. In this graph, the y-axis represents the number of DNA, while the x-axis represents the time. But don't get confused about the number of DNA and several chromosomes.